Great to be with you here today virtually at ATBC, and I'm going to expand the geographic scope of the symposium slightly by talking about biogeographic links from Amazonia and the Andes to the Mata Atlantica of the eastern coast of Brazil, depicted here by von Martius. So I think that all of you will know that the Mata Atlantica is the second biggest area of rainforest in Latin America, separated from Amazonia by dry diagonal of formations of savannas and tropical dry forests, the Caatinga and the Cerrado of Brazil. And the Mata Atlantica has a fantastically diverse flora, 15,000 flowering plant species, of which 50% are endemic. And that endemism, of course, reflects the isolation of the area. It reflects in situ speciation. But what I want to talk about today is not so much the isolation of the Mata Atlantica, but how it has connected in the past to Amazonia, because of course there are many species that are shared between these areas and many genera with shared distributions in both of those blocks of rainforest. Now, the narrative of connections between those areas has tended to emphasize specific windows of dispersal opportunity across the dry diagonal. And they're envisaged to happen at particular times when climate was perhaps wetter, allowing a greater connection of rainforest areas. And so you'll see here that one route, which is an older route, thought to have been opened in middle and late Miocene, is in the south. A more recent route is here up in the northeast across the coastal area of the Caatinga dry forest, Pliocene, Pleistocene. And in this very good review by Lado and Colley, the width of the arrows represents their view of the perceived importance of these dispersal routes. And there's another, seem to be less important, these thin arrows, which represents a possible dispersal route through gallery forests, which are particularly prevalent in the Brazilian Cerrado. Those are ribbons of rainforests along rivers. So what I want to do in this talk is use this genus, Inga, a mimosoid legume, as an example to look at Mata Atlantica biogeography. For those of you that don't know Inga, it's fantastically diverse, 300 species of trees. There's a lot of economic uses. So this is a little advert for a talk I'm going to give about use of Inga in silvopastures in a symposium tomorrow. Many trees of economic use for fuels, firewood, nitrogen fixation, etc. But it's a fantastic example to look at Mata Atlantica biogeography and links to Amazonia, because it has many species in the Amazon, nearly 150 in Andean region, 55 species in the Mata Atlantica. And if you look at that map, the dots in the dry diagonal represent species that are found in gallery forests in those drier areas. And it's really important to emphasize that Inga species are rainforest species. They don't grow in the fire-prone savannas, the Cerrados of central Brazil, or the Caatinga dry forest. And using a phylogenetic approach, I'm going to address two questions. One about whether there's a predominant direction of dispersal from between Amazonia and the Mata Atlantica. Another way of framing that is there an ancestral area for Inga? And more especially, I'm going to ask, are dispersal events clustered in time matching some of those proposed windows of Mata Atlantica Amazonia connections? I'm going to do that with what I think is some really pretty good sampling for such a species-rich genus. We've got 127 of the 145 Amazonian species and around 60%, 34 of the Mata Atlantica species. This is sampling carried out by a big team over more than 2 decades in many locations. And the data I'm going to show you are based on targeted enrichment of many nuclear genes. And what that next generation sequencing approach has allowed us to do for the first time in Inga is resolve the relationships really nicely of closely related species. And even as we showed in this 2015 paper, and it's important in the context of the study I'm going to tell you, is it can resolve relationships of populations within species. So the data are from 110 loci. So put together, that's more than 1 million base pairs of sequence. That data was all gathered by James Nichols here in labs in Edinburgh. The analyses are done by Catherine Kidner and Oriane Loiseau. And I'm going to show you dated phylogenies from work in progress by Jens Rinkelberg. So going straight to the data, this tree I'm showing you here is based upon all individuals in our analyses. We have multiple individuals per species in many cases. We've also got analyses of a species tree using coalescent approaches where we just have one representative per species. So I'm presenting results actually from 2 analyses through this talk. Many people would regard the species tree based upon coalescent approaches to be a more accurate phylogenetic estimate. And if you look at this tree, you'll see, thinking first about the question of direction of dispersal, that blue is coloured at the base of the tree. And we have red scattered throughout the tips. Red is Mata Atlantica. This is consistent with Amazonia being the ancestral area for Inga, and numerous dispersals happening from there to the Mata Atlantica. And in that individual level phylogeny, we see 18 colonizations from the Amazon to the Mata Atlantica and just 1 in reverse. In the species level phylogeny, 20 dispersals from Amazon to Mata Atlantica. So to show you a little more detail so you can understand what's going on, we have some examples where we have small clades of Mata Atlantica species, so evidence for in situ diversification. There's one at the top of this inset there, Inga sessilis in the beautiful photo on the top right, and its close relative Inga subnuda. But we also have examples of species that are widespread and found in both Amazonia and Mata Atlantica. And here we can see for Inga marginata clear derivation of the Mata Atlantica populations in brown from Amazonian populations. So again, clear evidence for that directionality of dispersal. But the main emphasis in this study is really to ask, did this dispersal happen at specific times? And these analyses I'm showing you here, led by Carl Dexter, have used a null model approach. What we've done is randomly scattered dispersal events across our 2 phylogenies and ask how those differ from the observed dispersal events. And you see that they don't really differ. So what we're seeing is that these 18 to 20 migrations are apparently stochastic and continuous throughout Inga's history, and that dry diagonal, ecologically so different, doesn't seem to be much of a barrier for the rainforest genus Inga. Now, legumes are dispersible. I spent a lot of my career looking at the biogeography of legumes. There's good evidence that through their evolutionary history, they, for example, cross major oceans like the Atlantic. But Carl Dexter showed in this paper that what goes for legumes may go for other genera of the Amazonian trees. So in that study, as well as Inga, we looked at Schwartzia, another legume, but also Burseraceae, and also Quateria in Ananaceae. And what we were looking at was the phylogenetic composition of local communities of these species. And what Kyle's analysis showed, again using a random null model approach, was that in Amazonia in particular, the phylogenetic composition of those communities, their phylogenetic diversity, was essentially random, that those communities represented a random draw of species, presumably by dispersal from a regional Amazonian meta community. So Inga and other genera are dispersible. And in the case of the Mata Atlantica, it seems that it has been able to cross the dry diagonal many times. And I think that that may represent dispersal through those thin blue arrows here of the gallery forests, which presumably, as long as the rivers are flowing, have been available constantly for migration. And I rather like the fact that we've been able to reach this conclusion because it was pointed out a long time ago in the mid-1990s by Ari Oliveira Filho and Jimmy Ratter. Ari was working as a postdoc at the Royal Botanic Garden Edinburgh when I started working there in the 1990s. And it's nice to see the importance of their study re-emphasized here. But I think there are some broader conclusions. And they are that I see that fundamentally a lot of biogeography in Latin America still is essentially grounded in a cladistic vicariance-based philosophy, in that it emphasizes seeking the effects of specific climatic or geological events. But it seems to me that for rainforest trees, that those events have often been overwritten by dispersal. It's not they have no effect, but their effect is hard to discern. And that has also been concluded for tropical birds in Amazonia by Smith et al. In that Nature paper. So I think that we should be thinking more about dispersal-based biogeographic models, where that dispersal is mediated by ecological traits. In other words, as a lineage, can you survive the drought of the dry diagonal or the cold of the high Andes? So I'd like to acknowledge, of course, we have to acknowledge our funders. So in this case, particularly a US National Science Foundation Dimensions Biodiversity Grant led by Phyllis Coley and Tom Kersar, and also some UK funding. I'd like to thank my collaborators. And in particular, I'd like to acknowledge two people, Tom Kersar and Jimmy Ratter, who passed away in the last few years. I think many of you will know Tom, few of you will know Jimmy, who was my first boss and manager and a great inspiration to me. These both were people who were models of generous, gentle mentorship, especially of Latin American colleagues and students. And it's nice to dedicate this talk to them. And thank you for listening.